start from a FASTQ file, a clean FASTQ file. And we're going to transform this FASTQ file by doing the mapping into a BUM or a SUM file that also contains the results of the alignment. We need a reference, so what are we going to map to? And once we have the reference, we can do the mapping, and you will see that for RNA-seq reads, there are special mappers for that. So, for instance, STAR, HiSET, Callisto, and Salmon. I will come back on that. So what do you map to? What is the reference? So in most cases, not always, but in most cases, you're going to map to the genome sequence, so the full genome sequence. This is an example for the human genome. That's a pretty large thing to map to. In that genome sequence, there are, for human, 20,000 genes annotated, and they only make up 2% of the complete genome. So that might seem a bit of an overkill, mapping your reads to the genome while you already know that they will only map to 2% of that whole sequence. That is why there are also mappers that will simply map to the transcriptome. I will come back on that. What is important also for RNA-seq data, so there are at this moment 20,000 genes, protein coding genes annotated in human, but these 20,000 genes give rise to more than 200,000 different transcripts. So every gene, or almost every gene, will encode many different transcripts. They're also called isoforms or splice variants. Okay, so this is something that, especially in higher eukaryotes, you might also be interested in. You can look at gene expression on the level of the genes, but you can also look at gene expression on the level of the transcripts. Okay, so what do you need for mapping RNA-seq data? So let's assume that we're going to map to the genome, as most mappers do. So if you map to the genome, you need the genome sequence, which is one or multiple huge FASTA files. If you want to map to these FASTA files, these huge files, if you want to know where your reads are located here in this file, you need to have an index. Yeah? That makes the mapping more efficient. That makes it easier to find your reads in this huge file. Now, on top of that, for RNA-seq, you need more than just that. You also need to know the annotation. You need to know where the transcripts are located in this FASTA file, because this is where your reads will map. They will, almost all of them will map to the transcripts. So on top of this FASTA file, you also need a GTF file. So this is a text file that contains the annotation, the location of the introns, the exons, the UTRs, well, the transcripts, as you can see here. So you see here, transcript is located here on chromosome 21 and belongs to this gene. And your RNA-seq mapper needs to know that, because this is where you expect that your reads will map. Where do you get your GTF files? You get your GTF file from the same website where you get your FASTA file. So if you get your FASTA from Ensembl, you will also get your GTF file from Ensembl.